Assalamu alaikum and good evening. My name is Atiya Hakim and I belong in the group Clover. And today we are going to talk about microarrays. Microarrays is a laboratory tool that is used in the lab so that we know about gene expression in particular organisms. Microarrays are basically a tray that has many labeled DNA probes. The DNA are of known sequences and we release the mRNA strands onto the microarray so that they hybridize with the DNA probes. Once the mRNA has hybridized with the DNA probe, we are certain of which protein exactly the mRNA is coding for, and therefore we also know how much of this protein that the particular cell or the particular organism expresses. To exactly know how microarrays work, we have to know about the central dogma of biology. The central dogma of biology is basically how the cell gets this information and conveys this information into the extracellular environment. It starts off with DNA replication, and once the DNA has replicated, we have the DNA going through transcription, whereby the nitrogenous bases, each three nitrogenous sequence in a DNA codes for an amino acid. This information is then transferred onto an mRNA strand, which is a single strand. The mRNA strand is then read by the ribosomes and with the help of tRNA strings together molecules of amino acids to form proteins, which is then released. Therefore, we know that mRNA is a good indicator of the gene expression of a particular cell or organism. Going down to the history of microarrays, we can talk about the first known molecular hybridization of DNA by Michael Grunstein and David Hognis. In 1975, these two scientists performed an experiment whereby they had a plate of E. coli bacteria and they blotted this plate onto a nitrocellulose membrane. They then released labeled DNA probes onto this membrane and they saw that the DNA hybridized with certain molecules, the mRNA. Taking this idea, Gergen et al. Then produced multiple plates of agar which were replicated to produce arrays through the use of mechanical pin device for placing samples in well microplates. This allowed for the production of arrays over 1,000 different bacterial clones. Basically, that Gergen et al. Managed to develop a method to hybridize many mRNA molecules on one plate. In 1994, Affymetrix, which is part of a company known as Thermo Fisher Scientific, released its first commercialized microarray known as the gene chip. The gene chip is a very small device that has thousands of DNA probes, labeled DNA probes that can also glow when they hybridize with the mRNA. Although in 1997, Stanford University was the first university to actually analyze the gene expression of an entire organism, of the entire DNA of an organism, of yeast in particular. They used a microarray for this, but they did not commercialize the microarray that they used, but rather they released instructions on how to assemble the microarray. My teammates will further talk about the different advantages, disadvantages, and many other facts about microarrays.
The principle of DNA microarray technique. The principle of DNA microarrays lies on the hybridization between the nucleic acid strands. The property of complementary nucleic acid sequences is to specifically pair with each other by forming hydrogen bonds between complementary nucleotide base pairs. For this, samples are labeled using fluorescent dyes. At least two samples are hybridized to chip. Complementary nucleic acid sequences between the sample and the probe attached on the chip get paired via hydrogen bonds. The non-specific bonding sequences that remain unattached and will be washed out during the washing step of the process. Fluorescently labeled target sequences that bind to a probe sequence will generate a signal. The signal actually depends on the hybridization conditions, for example, the temperature, washing after hybridization, etc. For the steps in DNA microarray, the first step, which is collection of samples, the sample may be a cell or tissue of the organism that we wish to conduct the study on. Two types of samples are collected, the healthy cells and the infected or tumor cells. These are for comparison and to obtain the results. The second step, which is isolation of mRNA, RNA is extracted from the sample using a column or solvent like phenylchloroform. From the extracted RNA, mRNA is separated, leaving behind rRNA and tRNA. As this is because mRNA has a poly A tail. Column bits with poly T tails are used to bind mRNA. After the extraction, the column is rinsed with buffer to isolate mRNA from the bits. Thirdly, the creation of labeled cDNA. To create cDNA, complementary DNA strand, reverse transcription of the mRNA is done. Both samples are then incorporated with different fluorescent dyes to produce fluorescent cDNA strands. This will help in distinguishing the sample category of the cDNAs. Hybridization. The labeled cDNAs from both samples are placed in the DNA microarray so that each cDNA gets hybridized to its complementary strand. They are also thoroughly washed to remove unbounded sequences. Next, data collection and analysis. The collection of data is done by using a microarray scanner. This scanner consists of a laser, a computer, and a camera. The laser excites fluorescence of the cDNA, generating signals. When the laser scans the array, the camera records the image produced. Then the computer stores the data and provides the result immediately. The data then will be analyzed.
So now I'm going to discuss about uses and application of microarray. So first, there are common types of microarray, which is the protein, tissue, cellular, antibody, and also DNA microarray. So first one will be protein microarray, which expresses the protein and interaction profiling. The next will be the tissue microarray, which compares histological section from unique tissues or tumors. Cellular microarray study the cell responses, while antibody microarray detect antigens and many diagnostics applications. Finally, DNA microarray measure the gene activity and also for genotyping. So there are more applications of microarray in this new technology world, but there are a few I would like to say, which is this transcription factor finding analysis, data standard and data exchange, genotyping, and also gene expression analysis. I would like to discuss three out of it, which is data standard and data exchange. It used to allow the vast amount of complex digital data, allow to access a raw and processed data to perform analysis. The FGED created MIAMI, which is the minimum information about a microarray experiment standards. So it is used for the description of microarray experiment and for the exchange of microarray data. So genotyping. So microarray creates genotyping platform for single nucleotide polymorphism. There are few alternative approaches that have been used to detect it. So these assays, this SNP for the assay are selected to be in between restriction sites that are less than 1 kilobyte apart. So this corresponding increase in signal to noise on the array. So these are the alternatives, which is allele description by hybridization, Illumina's Golden Gate assay, a rate primer extension, and also Illumina's Infinium assay. So the last one will be gene expression analysis. So it has been used to measure gene expression, which two most frequently used methods, which are incorporation of fluorescently labeled nucleotides in the cRNA or cDNA synthesis step, and incorporation of biotin labeled nucleotide in cRNA synthesis step. So these are the simplified methods, which is population of cell of interest are selected, and then it will be isolated, and then enrichment for mRNA. It will be amplified, that is an optional step, and then it will convert it to the two frequent step that I stated earlier, which is the fluorescently labeled cDNA or cRNA, and then biotin labeled cRNA or directly labeled with RNA fluorescent text. So it will undergo hybridization to array and wash, and it will undergo staining process to be scanned the gene expression. That's all.
Hello, my name is Nurin Fatini Afrina Mitifari and my number matrix is 17121332 or IIB 180062. So, I will be presenting on the research used on microarray. We know that we use microarray to determine which genes are expressed by the cell or tissue. So let's go on with this research on ovarian cancer cells. Ovarian cancer is the leading cause of death among women from gynecologic malignancies. To improve survival, it will be desirable to develop tumor markers that could be used to detect the early stages of the disease. So far, the researchers only managed to identify only a few genes in the last few years. In the research, they use microarray to identify genes that are differently overexpressed in ovarian cancer cells using RNA pulled from several healthy HOSE cell cultures and ovarian cancer lines. A total of 30 genes were identified. The genes then were further confirmed by the real-time RT-PCR. By using this, they managed to identify genes that either overexpress in all cancer cell lines or in only some cancer cell lines. Identifying and characterizing these genes will allow them to be used in diagnosis, prognosis, anti-cancer therapy, and molecular classification of ovarian cells.
Hello everyone, my name is Karishwini, matrix number SIO180009. So I would be talking about the advantages and disadvantages of microarray. So let's get into it. So the first advantage would be it contains many tissue specimens. Second would be for cancer research purpose. Microarray, some of the microarrays are said to have lower cost. And lastly, the speed, specificity and reproducibility rates are high. Okay, so why do I say the last one is because speed in terms of generating the array is the main advantage, because spotting the DNA onto the chip requires only the DNA of interest to be known. Therefore, no extra time would be spent in handling cDNA resources, such as the preparation and accurate determination of handling bacterial clones, PCR products or cDNAs, thus reducing the likelihood of contamination and mix-up. So this picture shows that it is a typical tissue microarray. It consists of paraffin blocks in which up to 1000 separate tissue cores are assembled in array fashion to allow multiplex histological analysis. This is one of the advantages, because it contains many tissue specimens to increase number of parameters that can be analyzed and giving a high level of result. So these two pictures down here shows the normal and multi-tumor microarray. So these are the different types of devices used to identify cancer cell lines. So let's go on to the disadvantages now. So there are 4 disadvantages. The first one would be cross hybridization. Second would be the cost. The third would be the decreased sensitivity, and last but not least is microarray analysis will not detect certain chromosome rearrangement. Okay, let me explain a bit about cross hybridization first. During microarray experiments in the laboratory, sequence homologies between clones representing different closely related members of the same gene family may result in a failure to specifically detect individual genes, and instead they may hybridize to spot design to detect transcript of a different gene. This phenomenon is known as cross hybridization. Okay, some microarray cost more than the rest because some requires more materials to carry out their specificity. Okay, now lastly, the fourth one is microarray analysis will not detect certain chromosome rearrangements such as balance, translocation and inversion, because although there has been an exchange of DNA, there is no net gain or loss of DNA detectable by microarrays, and many research has been done to prove this point. Okay, so this is a picture of the cDNA microarray technique. It's one of the disadvantages because the short sequences used on the array confer high specificity, but they may have decreased sensitivity or binding. Okay, so that's all from me about advantages and disadvantages of microarray. Thank you.